Good morning, everyone. I hope you're staying nice and dry at this time. It's such an ugly morning out there, so. Okay, well, typically on days where we have ugly weather, attendance is down in the class, so hopefully those that aren't with us this morning are not having some kind of problem with the water. So welcome back. Hope you're doing well. It's the kind of day you just want to stay inside and watch a movie or something like that. So let's see what we can do. We need to finish chapter 3. Okay, so we had some really important topics this morning. Chapter 3 is just a huge chapter with a lot of important content. Before we do you have any questions that you would like to ask? All right, so where I'm going to begin my coverage in chapter 3 today is talking about the broad topic of protein synthesis, which starts basically on page 93 of your textbook. Some of the figures that are relevant for this conversation include figure 3, 3.26, 3.27, 3.28, 3.29. Okay, so in that area of your digital textbook is what I'm going to be talking about. And so this is one of those big areas where it's probably going to be beneficial if you have some visualization before we talk about it. So I'm going to show you an animation on the process of protein synthesis, and then we're going to take some notes on it, walk you through it, and help you to understand. Okay, so let me share my screen with you. Okay, and you will notice that I already have 3 videos listed in today's notes. These videos will help you during your private study time. I'm going to show you the first video at this time from YouTube. It's actually, I believe, a McGraw-Hill video that's been posted on YouTube. So let's cue this up and we'll get started this morning. Here we go. Enjoy, folks. Here we go.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into 2 processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly. such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
Okay, folks. So that's a pretty involved animation there, but that's going to get our conversation started this morning. So we have lots to do. Would you have any questions you'd like to ask simply from having seen the video one time? Okay, so like most topics we talk about in this course, we do enough to whet your appetite and dip your toes into the water. You're not going to be an expert on genetics by taking this course, right? You need to study genetics for many years to really understand it. But let's give you some of the basic ideas here. So let me start with this. Do you remember last class, we were talking about the different transport processes? And we made this cartoon drawing in your notes where we explain glucose moving out of the bloodstream into the interstitial fluid and then into the pancreatic cell. And then we said the pancreatic cell would respond to the presence of glucose by making insulin. Well, insulin is a protein. And last class we said we're not going to explain at that moment how insulin was made. The video you just saw, is how insulin is made in the pancreatic cell. This is how all proteins are made. So the YouTube video above explains how proteins like insulin are made, okay? Your body relies on the production of proteins to do many, many tasks. So this is a really important concept. The topic of protein synthesis, the process of making a protein follows a multi-step process, a multi-step pathway, which is known by another phrase. It's known as gene expression, okay? So on page 93 of your book, where you have the large heading of protein synthesis, in the second paragraph, you see that bold-faced term in your digital textbook of gene expression. Okay, so the process used to manufacture proteins is known as gene expression. Okay, we are expressing the information of a gene. We are using the information of a gene. We are decoding the information of a gene in order to enable the cell to manufacture something, okay? All right, so I'm going to give you a couple diagrams here, a couple flow charts where we describe the process of gene expression, okay? Gene expression as a process can be summarized by a flow chart known as the central dogma. The central dogma. Okay, the central dogma is a pathway of steps leading to the production of a protein. Okay, now I'm going to express this idea two different ways to you. Okay. So I'm going to give you the simple version of the central dogma first. Okay, here's the simple version. DNA is used as a template to make RNA, which is used as a template to make protein. That's basically what you saw in that 7-minute video that we started class with. How do we go from the instructions contained in the DNA to a protein like insulin or any other protein you might be interested in talking about? Okay, this is the simplistic version. Okay, now what I want to go to you is a more detailed version. And to do this, I'm going to go to my drawing tools here because it's just easier to convey some information in a handwritten form. Okay, so let's do this here. I'll use a nice dark blue here. Okay, so the more detailed version of the central dogma goes like this. DNA. Now, when we say DNA here, there's lots of things about DNA you don't know yet. But when you're producing a protein, the specific unit of instruction in the DNA, which contains the instructions to make the protein, is going to be known as the gene, right? So you basically have a gene here. The gene is made of a sequence of DNA nucleotides. Okay, that gene is then used by machinery inside the nucleus to manufacture what we will say at this point is immature RNA. Okay, that immature RNA is going to go through a chemical process known as RNA processing, where snippets of RNA that are not necessary are going to be removed. So we're going to go from immature RNA to mature RNA. Okay. The mature RNA is going to be exported out of the nucleus. Okay. Going to be exported out of the nucleus, where a ribosome, typically attached to the membrane of the endoplasmic reticulum, the rough ER, is going to take the information in the mature RNA, and it's going to build a polypeptide from it. The polypeptide is then going to be used to assemble a protein. And then even the protein might experience some processing, which is known as post-translational modification. To become a physiologically active protein. Okay, so you might ask yourself, which version of the central dogma chart do you prefer? You probably prefer the simple version, right? But it is not that simple in reality. So let's talk about a few ideas here explained in the detailed version of the central dogma. Okay. And so I'm going to switch to a different color here just to highlight some things. Okay. So the process of taking a gene and using it to manufacture a piece of immature RNA, that is known as transcription. Okay, and I will highlight for you that transcription as a process is illustrated in figure 3.27. Figure 3.27. Okay. The machinery of the cell that is capable of performing transcription. I mean, transcription as a process requires multiple molecules to happen. And we'll describe a little bit of it, but I want to give you the name of the major enzyme, the major molecule involved in the transcription process. Folks, the major enzyme that takes the DNA, the instructions of the gene, and uses those instructions to assemble immature RNA is known as RNA polymerase. Okay. And so I hope you can see when you look at figure 3.27 in your book, figure 3.27, I hope you can see picture B there. And in fact, you see RNA polymerase written out in that picture of your book. So the gene, which is made of DNA, is used as a template by RNA polymerase to perform the process of transcription to make immature RNA. Okay. Next, that immature RNA has to go through a process known as RNA processing, or simply processing for short. Okay. Your textbook does not have a separate figure for RNA processing. It does not have a separate figure. But what I want you to appreciate is that RNA processing is alluded to in the bottom half of figure 3.27, right? Figure 3.27 is showing you the process of transcription, but it is also hinting at the RNA processing towards the bottom of that image. Okay, so here's a question in the chat window. Okay, here's a question in the chat window. Here we go. What color is the molecule called a SNRP in figure 3.27? Okay, thank you, Lauren. Do the rest of you see it? Okay, I need you to be looking at your books. I need you to be focusing on this content. Folks, if you're looking at that green blob in figure 3.27, if you go to the right of the green blob, you should see a ribbon of RNA and then a bracket that says intron snipped and deleted. And if we go to the left of the green blob, you should see brackets which say exons spliced together. This is the RNA processing I'm talking about. That is in figure 3.27. Look at the green blob, the information content of the green blob and around the green blob. That is RNA processing. Okay. The name of the major machine involved in RNA processing is a SNRP, as I'm calling it. It's hard to say it, okay? The SNRPs are short for small nuclear ribonucleoproteins, which is a term in boldface in the writing of chapter 3, right around figure 3.27. Okay, so the machinery to do the RNA processing is the snRNP, which also has an alternative name. Sometimes it's simply called the spliceosome. Okay, so RNA processing is achieved through a machine called the SNRP. Okay, each of these steps we're going to focus on a little bit more detail in just a moment, but I'm just giving you the overview in the detailed version of the central dogma pathway. Okay, now it's important that you appreciate that everything we've talked about so far, okay, DNA to transcription to immature RNA, which is processed to mature RNA, all of this activity here that I'm starting to outline in green, all of this activity takes place inside the nucleus of a cell. All of that activity takes place inside the nucleus of the cell. The nucleus is an environment that is optimized for the process of manufacturing and processing the RNA. The machinery to do all of that is in the nucleus. Okay. So as we've indicated on your flow diagram here, okay, as we've indicated, the next step is going to be to take the mature RNA and export it from the nucleus. Okay, so let me just see here. I just want to see something. Okay. Question in the chat window. Question in the chat window. Here it comes. What part of the nucleus is used to export the mature RNA? I want you to take a look at figure 3.24. Take a look at 3.24 and tell me what part of the nucleus is used to remove the RNA from inside the nucleus to out into the cytoplasm. Okay. Rebecca, thanks for answering. You're the first person to answer, and your answer is correct. So good job. Okay. The nuclear pore, right? The RNA is going to be thread, like a thread and needle. It's going to be thread through the opening of the nuclear envelope, the nuclear membrane, called the nuclear pore. Okay. So we start to see how these processes relate to the actual anatomy of the cell. So export from the nucleus through the nuclear pore. Okay. So once the RNA is in the cytoplasm, okay, so it can be free floating in the cytoplasm or it can be shuffled into the endoplasmic reticulum. It happens a couple different ways. The next thing that's going to happen is that the polypeptide is manufactured. Okay, the polypeptide is going to be manufactured through a process known as translation. Okay, so I'm a little crimped here in my writing space, but right here is translation. Okay, so translation is going to be the process that is used to make the polypeptide. And the machinery to make the polypeptide is the ribosome. Okay, now you can see that your book goes into pretty good detail really about this process of translation. Figure 3.29, figure 3.29 is the picture for you. I'm not going to walk you through all those steps. I mean, I'm going to tell you a little bit about it, but you're going to need to spend some of your private study time to do this. Okay, so the mature RNA is transported out of the nucleus where some ribosomes present in the cytoplasm translate the RNA into polypeptide. A lot of people think that polypeptide and protein are the same thing. And you know what? They kind of are, but there is a subtle important distinction. Okay, when you have a polypeptide, you have a molecule that is kind of like a shoestring. And by a shoestring, I mean it's kind of limp and it's laying all over the place. It doesn't have much structure to it. So a polypeptide tends to be unstructured, meaning it does not have a defined shape. No defined shape. Okay, so the polypeptide is going to fold. Okay, there's going to be a folding process that takes place inside the cell. That folding process is not entirely understood by science. We don't entirely understand it, but I will say to you that sometimes the polypeptide is folded into its proper shape by the activity of special proteins called chaperones. Okay, chaperones would be the proteins that help the polypeptide adopt its proper folding, its proper geometry, its proper shape. Okay. So when you have an actual protein, you now have a defined shape. Okay, so if I were to use an analogy, a polypeptide would be like a shoestring from your shoe that you're just holding out between your fingers. Okay, it's just limp and loose. A protein would be when you take the shoestring and you tie it into a very nice bow. So polypeptide is just an open shoestring. Protein is a shoestring that's been folded into a bow and has a defined shape. Okay. Let me ask you this in the chat window. What does the property of shape define? We talked about this before in chapter 2. What does the property of shape define? Okay, thank you, Lauren. Sierra, good job. It defines the function. That's what I'm trying to get you to see. A polypeptide really does not have a function yet because it doesn't have a defined shape. Once it folds into its correct shape, which is a complicated process that is not entirely understood, once the polypeptide takes on the shape and becomes an actual protein, it now has function, or at least has the capability to potentially function. Okay, for some proteins, there will be additional modifications to the protein, which I'm defining as post-translational modifications. Post-translational modifications would include the addition of chemical tags to the protein, small groups of additional atoms, maybe methyl groups like CH3 groups or other things might be added to the amino acid backbone of the protein to give the protein its final physiologically active ability inside the cell. Okay? All right. Folks, do you have any questions at this time? If anything goes wrong with any step of this process, you can get a protein that is dead on arrival and has no physiological activity, or maybe a protein that doesn't ever even get manufactured or produced. And that would be the foundation for many disease processes, some of which we understand, some of which we do not understand, right? So, and just to complete our flow diagram here, you appreciate that everything outside the nucleus, right? Everything outside the nucleus that I'm highlighting in orange is happening at some location in the cytoplasm. Okay. All right. Gang, is there any questions you'd like to ask at this time? All right, so this gives you an overview of the process of gene expression, which is described as by this pathway called the central dogma, which is what the video you saw at the beginning of class was all about. I mean, that's what the video was trying to show you, is how these various processes take place. So what we're going to do next is break these things down. I'm going to pick certain things and talk a little bit about each thing, okay? All right, so let's talk about transcription a little bit, okay? And again, this can't be an exhaustive lecture. I mean, I could do a whole hour lecture just on transcription, but let's make some bullet points here for things that you should begin to appreciate. All right, so remember, transcription takes place in the nucleus. The name of the major enzyme that performs the transcription process. Okay. All right. I want to see if you can answer a question in the chat window. So we're telling you in your notes at this time that there's an enzyme in the nucleus called RNA polymerase, which is used to perform the process of transcription. In the chat window. How does the cell know how to manufacture RNA polymerase? How does the cell know how to manufacture it? What do you think, folks? How does the cell know how to manufacture RNA polymerase? Okay. All right. So I'm seeing some nice words come up in the chat window. Base pairing, promoter. That's kind of relevant. Yeah. Okay. So thanks for those that are attempting to answer this. What I'm trying to draw out of you is a realization that for the RNA polymerase enzyme to physically be present in the nucleus, that enzyme itself must have first been manufactured through the process of gene expression. So what I'm saying is the cell knows how to make RNA polymerase because in the DNA, there is an RNA polymerase gene. There is an RNA polymerase gene, which has the instructions that the cell decodes through the process of gene expression described by the central dogma pathway to manufacture that enzyme. So any protein you see in the cell, how did the cell know how to make it? It knew how to make it because there's a gene in the DNA which tells the cell how to make this thing. Okay, it's kind of like a cookbook you have at home where, you know, there's a recipe for barbecue chicken, there's a recipe for angel food cake. How do you know how to make these things? Well, you decode the information in the recipe, right? The recipe is the gene. Where's the gene? It's in the book. It's in the DNA. Okay. All right. So the process of transcription works something like this. Okay. Let's talk about this. Okay, so when DNA is used as a template to make RNA, there are rules for how the nucleotides are matched together. Okay, so if I have a strand of DNA and I'm going to make RNA from it, here's what the rules kind of are. Okay. If I have adenine, okay, adenine is a nucleotide. It's actually the nitrogen base part. Okay, if I have adenine, which I'll abbreviate as an A in the DNA chain, then RNA polymerase will put a uracil, meaning a U, in the RNA. if DNA has a thymine nitrogen base, abbreviated T, then in the RNA polymerase will put an adenine, an A. If there is a cytosine in the DNA, then RNA polymerase will put a guanine in the RNA. If there is a guanine in the DNA, then RNA polymerase will put a cytosine in the RNA. Okay, so let's do this. Let's put this into your notes here. Okay, so if I have this DNA template, if I have this DNA template, type into the chat window what the RNA would be. Type into the chat window what the RNA would be. Tony, I see your comment. I'll address that in just a second. What is the RNA sequence that will be made from this DNA template? What would the sequence be? Okay, right, now we're getting some good answers here. Okay, just look at the chat window and you can see a variety of responses that are generally correct. Okay, very nicely done, right? The answer is, reading from left to right, it would be AU GGG CCAAU. Very nicely done. Okay, I'm going to put this in a different font at a different color at the moment, just so you can see it separately. Okay, AU GGG CCAAU. Okay, that would be the sequence that's made from the DNA template. Okay, good job. All right, so okay, so is that making sense, folks? Are you okay? All right, so let me go back to Tony's question. Tony asked in the chat window, could you repeat that, how it knows when to manufacture it? So Tony, I'm assuming you're talking about RNA polymerase. Is that what you're talking about? Okay, so Tony, I did not answer the question that you just asked. What was answered was, how does the cell know how to make the RNA polymerase? That is a very different question from how does the cell know when to make the RNA polymerase, okay? Your question is a very good question, but it's a deep question that would take a long time to answer. Let me say it this way. A cell knows how to make the RNA polymerase because the instructions of how to make it are found in the DNA as an RNA polymerase gene. How does the cell know when to make RNA polymerase? That is a really powerful and important question. And I would say this at this moment. The process of making proteins is happening all the time inside the cell, that basically for RNA polymerase, how does it know when to make the protein? It makes the enzyme RNA polymerase all the time, constantly, because the cell never stops making protein. Okay. Okay, other questions? Okay, so these are some basic talking points about transcription. All right. And so let me continue here. I want you to appreciate there's actually multiple types of RNA that get manufactured inside cells. This in itself could be another lecture, which I don't have time to give you, but let me hint at some of this complexity. So the first type of RNA we'll introduce you to is something known as messenger RNA. This is mRNA. Okay, up here in the flow chart where you're taking DNA to RNA to protein, that's the form of RNA that I was hinting at. Okay, so messenger RNA. This is the type of RNA that is being described in the central dogma flow chart above in your notes. Okay. The messenger RNA is going to be the template for a ribosome. However, there are other forms of RNA that get made and they have different functions in a cell. There is also something known as transfer RNA, or tRNA for short. The tRNA is a molecule that participates in the production of protein by being a delivery guy for amino acids, right? Think about transfer RNA as the Federal Express, the FedEx guy delivering a package of amino acids to your home if your home were a ribosome. So the job of transfer RNA is to bring amino acids to a ribosome to help the process of making a protein happen. Okay, this is a delivery molecule which delivers RNA to ribosomes to enable the process of translation, which we're going to be talking about soon. Okay. There's a third form of RNA inside the cell, which is ribosomal RNA. This is known as rRNA. The ribosomal RNA is significant because the ribosomal RNA, two things I need to tell you here. The ribosomal RNA is actually a physical component of the ribosome. Now, if you ask yourself, well, what is a ribosome made up of? The answer would be a variety of different proteins and a variety of different types of RNA that are known as ribosomal RNA. So the ribosomal RNA is a structural component of ribosomes. And B, the other important part, which, well, it's important to geneticists, it may not be important to A&P students. The other important part is that ribosomal RNA is the part of the ribosome that is capable of linking amino acids together. What I'm telling you is the ribosomal RNA is the catalytic component of a ribosome. It is able to join amino acids together by forming what is called a peptide bond, which is a special type of covalent bond that you learned about in chapter 2. Responsible for joining, catalyzing amino acids together by creating peptide bonds. Okay. All right. So the process of transcription is used to make messenger RNA, transfer RNA, and ribosomal RNA. These different forms of RNA can be used for a variety of purposes, but generally speaking, they're somehow involved in the process of supporting translation, the ultimate production of a protein. Okay. Any questions right now? Okay. All right, let's talk about RNA processing a little bit. This would be number 5. Okay. RNA processing takes place inside the nucleus. It uses a machine which is either referred to as the spliceosome or SNRP. They are equivalent terms. Okay. This process is performed by a multi-component machine called a SNRNP or spliceosome. Okay, these are synonyms. SNRP and spliceosomes are synonyms. They mean the same thing. Okay. And so the basic process is this, that when the immature RNA is made, there will be stretches called introns and stretches called exons. Okay. So what has to happen is the introns are spliced out of the immature RNA. Introns are removed from the immature RNA to form mature RNA. When the introns are spliced out of the RNA, the exons are glued together. Okay, so in a very simplistic and quick diagram, okay, when the immature RNA is manufactured, it might look something like this. Okay, hold on. All right, so if I have immature RNA, it might look like this. Okay, so I'm using color coding to suggest that there's different sections of the same physically continuous molecule. So I'd have immature RNA here. And I could have exon 1, intron 1, exon 2, intron 2, exon 3. Then I'm almost out of space here, but what will happen is the SNRP will get busy doing its biochemical activity here. And you will literally remove the introns using chemical processes that we're not going to try to simulate, but you're going to remove the introns and end up with a molecule that is now physically shorter in length because you've removed the introns. So we now have, and just to highlight it, we now have, right, we now have exon 1, exon 2, and exon 3 glued together. This is the mature RNA here. And this will go on to be translated by the ribosome. Okay. All right. So for a very long time, in fact, the way I was taught this topic some decades ago, okay, so I'm ancient folks, some decades ago, I was taught that when the introns are removed, that they're just chewed up by the cell, and they're basically waste and junk products that the cell doesn't concern itself with anymore. It's just garbage, throw it out. And what we've learned in the few decades since I've been sitting in your seat is that is really a false statement. The introns are removed, but they're not junk at all, and they're very rarely just thrown out the window, so to speak. The introns themselves, while they may not be used to actually make a protein, the introns are used to regulate the expression of genes, to control, in Tony's question, when a gene should be on or when a gene should be off. So even though the introns are removed, they're not thrown out. Okay, let me type this up here. That, and let me hint at this, introns have regulatory capacity to control when genes are turned off and on, control okay. All right, so that would be a conversation for probably another course, but I'm just wanting to hint at you just the, if you look at everything you're being asked to study, number one, it's intense and sometimes overwhelming and scary. But when you can, for a moment, forget that you're going to have to take an exam on this, you can sit back and marvel at what Mother Nature has created. It's just an amazing, beautiful thing of what it does. I mean, how does all of this translate into the fact that you and I are alive and we are consciously aware that we're alive? We know we exist. We know we're alive. We're inhaling air. We're thinking about how we're going to entertain ourselves this weekend, whatever it might be. Okay, thoughts, questions about RNA processing? Okay. See if I can get my next page in here. Okay, number 6, translation. Okay, translation is the topic of figure 329, page 95 in my physical textbook. Translation takes place in the cytoplasm, possibly in the endoplasmic reticulum. Okay, what I'm saying is there are ribosomes that are attached to the endoplasmic reticulum. So in that case, the translation would happen in the endoplasmic reticulum. Of course, the endoplasmic reticulum is itself located in the cytoplasm. There are, however, other ribosomes that are not attached to the endoplasmic reticulum, that are simply floating around like beach balls bobbing in the water of the cytoplasm, and sometimes they also translate. So however we think about this, translation is always taking place inside the cytoplasm in some capacity. Okay, so translation takes place in the cytoplasm. The machine that performs translation is the ribosome. Okay, and I'm going to ask you to spend some time looking at all the pictures of figure 329. I just don't have time to go blow by blow on this, but we expect that you have at least read this during your private study time. The basic idea for translation is, well, let me give you some additional background information. Okay. Within the mature messenger RNA, any 3 groups of nucleotides, any any group of 3 continuous nucleotides. That means one nucleotide followed by another nucleotide followed by another nucleotide. Any group of 3 continuous nucleotides is called a codon. Okay, it's called a codon. Each codon tells the ribosome what amino acid should be placed into the chain of polypeptide that's currently being manufactured. So each codon specifies 1 amino acid. Okay. Now in the video, you may have taken notice that when they were talking about translation, they flashed up a genetic code table. It was a fairly complicated looking thing. Let me see if I can bring up a quick image here with genetic code table. Okay, so one of these beasties, right? This is the genetic code table. All right, any 3 continuous nucleotides like CCU, CCC, CCA, CCG, all of these groups of 3 nucleotides, each group of 3 is a codon, and each one of them tells the ribosome that a proline amino acid should be placed into the protein. Okay, so this, you don't have to memorize this, but you need to be aware that this thing exists. Okay, so each codon specifies 1 amino acid. Okay, in other words, this tells the ribosome where to link specific amino acids together. So the basic idea would be this. Let's say we have, let's say that we have a stretch of amino acids, excuse me, a stretch of codons. I'm going to do it this way. CAC, CAU, CAC, ACG. Let's see what that does. So I'm going to go to my genetic code table and look for CAC. CAC, that's histidine. Okay, so I need to put in histidine, right? So histidine goes into that position. Okay, let me separate these out by more space so we can write. All right, next I need CAU. All right, CAU. Oh, that's also histidine. Okay. Well, CAC is the same thing we started with. That's histidine. Let's see what ACG is. ACG, that's threonine. THR, threonine. Okay. All right, so the ribosome would interpret the CAC, CAU, CAC, ACG in the mature messenger RNA, and it would link histidine, 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 threonine, amino acids together, and specifically that sequence. Okay. All right. Folks, do you have any questions right now? Okay, so that's translation. Once translation has successfully occurred, okay, we're into the polypeptide. The polypeptide will fold and form the protein of a defined shape. There may be some additional chemical modifications that your book is not describing to you, but I'm telling you that they're there. And then we get the physiologically active protein. Okay, Is there any aspect of this central dogma chart you'd like to ask a question about? Okay, all right. Well then, folks, I'm going to roll forward here. I don't have a lot of time remaining on you, and today is the last day I'm going to spend time in chapter 3, right? We did 3 lectures on chapter 3. I haven't covered everything, right? But that's the way it is. We will start chapter 4 next week. What I'm going to do next is just get a couple notes into your notes here about cell reproduction. Okay. Cell reproduction. Okay. And the videos that I've highlighted at the top of your notes here are to help you understand these processes. I'm not going to have time to lecture on them in any great detail, but the videos will help you at least get started as you try to learn some of this content during your private study time. Okay, you know from reading or from other things you've seen and heard in your life that there's 2 major forms of cell reproduction for animal cells like our own cells. Those forms of reproduction are mitosis and meiosis, right? So your book does talk about these things, right? Pages 96, 97, 98, 99, 100, 101, 102, 103. My goodness, it's a lot. Okay. So even though I don't have the time to lecture on every piece of every chapter, my hope is that by having our conversation twice a week through lecture, you start to learn how to think about biology as you're learning it on your own at times. Okay. All right. So the goal of mitosis is to manufacture genetically identical cells. Okay. The goal of mitosis is to manufacture genetically identical cells. And so for that reason, okay, if you fall on cement and you scrape your knee open, right, and you get a little blood there, it hurts, right? But obviously that area of your body will scab over and your body will attempt to heal itself. And some of the healing will involve making new cells to replace the damaged tissue. Well, the new cells that are going to be made, they're going to be made by mitosis. And they are made in such a way that they should be identical to the previous cells that were damaged. Okay. So the goal of mitosis is to manufacture genetically identical cells. This will support healing of the body when it is damaged, and it will also support growth of the body. In other words, when you were 3 years old and you were 2 feet tall or whatever it was, you know, you're 2 feet tall when you're 3 years old, but when you're 18 years old, you're now 6 feet tall. As you grow up, you have to make new cells so that your body enlarges. Generally speaking, the majority of those new cells will be made by mitosis, and they will be genetically identical to the precursor cells that created them. Okay, so this is also used to grow your body from an infant to an adult. Okay. So the cells that get manufactured through mitosis will be genetically identical. And for the most part, they're going to be genetically identical body cells. That word body is a loaded word to a biologist. That I mean a somatic cell, which is to say a non-reproductive cell, which means that mitosis is going to create cells that have 46 chromosomes or 23 pairs of chromosomes. Cells made from mitosis will have 46 individual chromosomes or 23 pairs, half from your mother, half from your father. Okay. The goal of meiosis is different. Okay, the goal of meiosis is to manufacture reproductive cells, your sex cells. Okay, the goal of meiosis is to manufacture reproductive, meaning sex cells. I'm specifically talking about the process the body uses to manufacture the sperm in men and the egg in women. Okay. This process makes the male sperm. Okay. As you know, sexually mature males will make sperm every day. Typically on the order of 200 to 400 million sperm are made newly every day. Okay. This happens every day in sexually mature males. Okay. The process of meiosis in women is handled differently. When a bouncing baby girl is born, that bouncing baby girl is already born with all the immature egg cells she will ever need when she grows up and becomes sexually mature. And so the process of meiosis begins before birth in the female body to create immature egg cells. Okay. Okay. Those immature egg cells that a baby girl, an infant girl, are born with are made before the baby is born. And then they pretty much are dormant till the baby girl reaches puberty and becomes sexually mature. Once the girl becomes sexually mature, then as you go through your monthly menstrual cycle, that monthly event, eggs will mature. They will complete the maturation process going through meiosis 2. And, you know, if sexual activity is involved, potentially that egg cell will be fertilized. Okay. The egg cells become mature on a monthly basis when the female reaches reproductive maturity. Okay, so the whole goal of, I know I'm almost out of time, the whole goal of meiosis is to produce the sex cells, the sperm and the egg. When meiosis has completed its job, the sex cells that have resulted will have half the number of chromosomes, 23 individual chromosomes, not 46. Mature sex cells will have half the number of chromosomes, which is to say 23 individual chromosomes. And significantly about this, those chromosomes will be different from the parental cells that were used to make them. And that happens through a process of swapping segments of DNA around like you're shuffling a deck of cards. It's a process known as homologous recombination, where you take one piece of chromosome and you swap it with another piece of chromosome and you swap positions. So what I'm saying is the genetic material is swapped around to create different combinations of genes so that your children will not be a genetic clone of you, and you are not a genetic clone of your parents, right? You might have similar characteristics to your parents, but you are not a genetic clone of your parents. And that is largely due to the fact that the DNA gets switched around, it gets recombined during meiosis. The chromosomal composition is shuffled to intentionally create genetic variation so that our children are not copies of ourselves. Okay, we don't want that. Mother Nature doesn't want that. That would be boring. Okay, and it also would not be very healthy from a species perspective. Okay, folks, I'm out of time. Do you have any questions? Okay, so the most important thing immediately for you is that you get ready for your lab exam next week, right? That is so important. It's a big exam. It's worth a lot of credit to your final course grade. You really want to nail it really, really well. But you want to stay up with your lecture stuff because, you know, next week marks 2 weeks before your first lecture exam. So that's going to come up quick too. Do your best, take breaks, but you got to be studying. Folks, have a great weekend. Stay safe out there. See you later.
Thank you.
Thank you. Bye.
Bye-bye.